PhD candidate Julia Constance. I've been one of her supervisors and had the pleasure of supervising her in her project. Today she's going to talk about the main topics of her thesis, which are the threatened river sharks. She already has 2 of her chapters of her thesis that are published, one that's been submitted and almost hopefully accepted.
Now.
And she also has submitted her thesis, so she's also just waiting revisions on that. And so I won't go into the details because this is what her talk is about. Thank you.
Thanks, Erica.
Hello everyone, thank you for having me here today. Very long title name, but essentially we're looking at aspects of river shark biology and movement ecology, and essentially informing conservation management of these species through all of this research. So firstly, I would like to acknowledge the Larrakia people whose country I'm privileged to be presenting on today. I also acknowledge the several nations on which I live, work and conduct research, including the Larrakia, Woolna, Combudge, Murra, Yugal, and many other nations. I acknowledge the ongoing fight for rights over water and acknowledge the significant cultural importance of waterways. So elasmobranchs are a group of what we call cartilaginous fishes, and they're a highly diverse group of about 1,250 species globally. Unfortunately, today there is almost a third threatened with extinction, and this is primarily due to overfishing. So 100% of our threatened species are impacted by overfishing. And then the impacts of overfishing are then compounded by things like habitat loss, degradation, climate change, and pollution as well. So within our elasmobranchs, our sharks and rays, we have a group of 10 euryhaline species, which make up less than 1% of our elasmobranch diversity globally. And these euryhaline species are physiologically capable of occurring anywhere from freshwater through to marine, and they carry out critical parts of their life history in non-marine environments. So for example, using river systems to give birth, or using river systems as nursery areas for their young. So they're not restricted to fresh water, but they can go literally anywhere through those environments. So these guys, compared to 1/3 of all sharks and rays, 70% of those are threatened with extinction, and that is primarily due to most of these species occurring in the tropics, where we have much larger populations of humans. And they also live in river systems and near-shore habitats, so they're very easily accessible. So again, they're threatened with overfishing and also habitat loss and degradation. So it's not looking good for these guys. So we have 10 species of euryhaline sharks and rays globally. We are very lucky to have 5 of these in Northern Australia. We have the bull shark, northern river shark, speartooth shark, large tooth swordfish, and the freshwater whipray. When we look at our river sharks, these are in the family Carcharodontidae in the genus Glyphus. There are 3 species globally, so the northern river shark, the speartooth shark, and then there's also the Ganges river shark, which does not occur in Australia. These all live in small populations. So for example, the largest population of the speartooth shark occurs here in the Northern Territory in the Adelaide River. That's limited to around 900 mature individuals, so very small. They have patchy distributions, so they have very niche habitat requirements. So they're not found in every single river system. They're only found in certain river systems that have the right habitat for them. They have fine-scale population structuring. So between those river systems that they are found in, there is very low connectivity between those populations, because the mums, they will return to the river that they were born in to give birth to their pups. And so any dispersal that does happen between those populations is driven by a few males that might swim between. And then we have very conservative life history as well. So they're slow to mature, and they're long-lived, and they also don't produce many pups and not that regularly either. So all of these lead to these species being classified as threatened. So here are the 2 species that we get here in Northern Australia. We have the speartooth shark and the northern river shark. They're categorised or really distinguishable by their really tall second dorsal fin. It's a bit complicated in the coastals, but when you're in the river systems, this is how you're going to tell. And then to tell the 2 species apart, you're going to look at this waterline mark, which is where the grey goes from white to on the underside of the body, and the Speartooth Shark is right below the eye there. So as I said, both these species are listed as threatened species under both the EPBC Act and the IUCN Red List. So I had a lot of chapters going on in my thesis, so we're going to go through each one of these. So the first one was a review of the life history and ecology of both euryhaline and estuarine sharks and rays. Then we're looking at the Roper River and what sharks are in there with a range extension for the Speartooth Shark. Then we're going to use acoustic telemetry, which will provide mortality estimates, and then that allows us to look at movement ecology and space use for each species as well. So moving straight into our review of the life history and ecology of euryhaline and estuarine species. So I've already explained what the euryhaline species are. The estuarine species are a group of 19 sharks and rays, as categorised by Grant et al. In 2019 as well. And they can't go into fresh water, but they do spend a lot of their time in estuarine rivers. So we know that the estuarine and euryhaline species are disproportionately threatened, and we really need data to be able to effectively conserve these species in their habitats. So we need life history data that can be age and growth and maturity, growth and age at maturity, also natural mortality, reproductive biology, how many pups are they giving birth to, how regularly, what's their gestation length, ecology, so movements over short and long term, and habitat use, and then population structure, again, looking at how connected those populations are. And so for 29 species, I looked at whether we have this data available. So to do that, I looked at over 300 journal articles. Will it go? Over 300 journal articles. There we go. 300 journal articles, species assessments and reports. And I threw it all into these giant tables. So for each species, and then we've got a whole bunch of parameters that each species looked at. Essentially, where there's big gaps, we have literally no information for that. So then I took 14 key parameters and I did a gap analysis. So for each species and each parameter, I looked at whether that parameter was well understood, whether it had a little bit of basic information, or whether it was poorly understood. And so you can see that there's a whole bunch of red going on for these guys. So we have major knowledge gaps for our euryhaline and estuarine species. Only 2 species are well known. These are our bull shark and the large tooth storefish. And they have information on over 80% of the gap analysis parameters. But even for these species, despite their almost global distribution, pretty much all of that data comes from 3 regions, Australia, the US, and Nicaragua. So that's potentially masking any regional differences in their biology, which is pretty well known that we do have regional differences in sharks and rays. 9 species are virtually unknown, with pretty much no information on over 80% of the gap analysis parameters. And the broad-nosed wedgefish, that guy, Rhynchobatus spengeri, there's no information on all but 1 of the gap analysis parameters for that species, even though it's listed as critically endangered. So we're lacking key data for management of these threatened species. Most of our research to date has focused on charismatic and commercially important species, but many of the euryhaline and estuarine species are rare, and the cost and logistics of doing that research has contributed to the lack of data. Rays represent the bulk of our euryhaline and estuarine species, and are at a higher risk of extinction than sharks. And they also form a larger proportion of bycatch and catch when we're talking about rays. Yet most of our research has focused on sharks. And although our rays of the family Dasyatidae, so our stingrays, make up the majority of our estuarine and euryhaline species, most of our research is focused on the swordfishes. Not to say that that's not important, but we do need to be sharing those resources around. Some of these gaps are also caused by taxonomic issues. So some of these species are difficult to distinguish, and they've also recently been delineated or described as a new species. But despite these difficulties, these species are ecologically unique. and highly threatened. So we do need to urgently put resources into getting this data. This review also highlighted key gaps in river shark science as well, a few of which will be covered through the following research. So looking at the Roper River, understanding the distribution of animals through time and through space is really important for conservation and management. We obviously need to know where they are to make sure that our resources are being directed to the right places. Northern Australia is considered a lifeboat region for many of the world's most threatened sharks and rays, and that's primarily driven by our small human populations, our limited development, and we have huge areas of land that are completely remote and untouched, but they have been historically poorly surveyed. So this was published back last year, and we went and had a look at what was in the Roper River. So here we have the Roper River. It's over in the Northern Territory's Gulf of Carpentaria. It's a very, very large river, and it is spring-fed, so it's perennial and it's forever flowing. So in 2016 and 2017, my supervisor Pete did surveys for sawfish upstream around Nooka and Roper Bar Crossing. And then I did surveys for river sharks in 2023 and 2024, closer down towards the mouth. And then I also did a literature review and data search for records of sharks and rays throughout the Roper River. So for this, we came across 5 species of sharks and rays. 4 of these are euryhaline species. So we have the bull shark, the speartooth shark, the large-tooth sawfish, and the freshwater wood ray. But we also found the giant guitarfish, or what people call the shovel-nosed ray, Glaucostegus typus, down towards the mouth, but in estuarine waters. So that was a bit of a shock. But we have the number recorded, their sizes, and the water quality parameters that they were found in. We have done one more survey since this was published. So we're up to 85 bull sharks, 105 speartooth sharks, and 18 freshwater wood rays. So if we look at where these were distributed along the Roper River, this is down towards the mouth of the river. The public boat ramp is about 15 kilometres upstream from the mouth of the river. And so we can see that we have quite a lot of, well, we have all of our species occurring in this area of the river. We have our giant guitarfishes close to the mouth there. We have our speartooth sharks, which are actually restricted to this area. So we didn't catch them further upstream than 66 kilometres. And then interestingly, our bull sharks in 2023, they were not found downstream of the hawk's nest there, but in 2024, they were absolutely everywhere. So it would be interesting to continue these surveys and figure out what is normal and what's not. When we look further upstream, we have the bull shark and the large-tooth sawfish, but the Yuggulmargi Rangers have also confirmed that we get freshwater wood rays all the way up to at least Gilpingan as well. So we've got large-tooth sawfish all the way up to Elsie National Park at Mataranka there, so that's nearly 400 kilometres upstream. And we also have records of sawfish way up outside the bounds of the map, 300 kilometres up the Biggetti or the Wilton River as well. So the Roper River is a very important river for our euryhaline species. It is a significant range extension for the speartooth shark, which from the Gulf of Carpentaria was only known from the Wenlock River in Queensland. It's an important reproductive area for all of these species, indicated by the presence of early life stages, so neonates and juveniles, and also pregnant females. We have elasmobranchs occurring to almost 400 kilometres upstream. And this research also provided new insights into freshwater wood ray biology. These guys are very poorly known. So we got a new maximum size, we got some reproductive seasonality information and new sizes at maturity as well. So you can see this girl here, she's got a very, very fat belly. She is pregnant, and we caught quite a few pregnant females in September. So the Roper River provides critical habitat for threatened euryhaline species. And more research is required to understand the life history and ecology of species occurring in that river, their habitat requirements and use within the river as well, and their connectivity with other populations. So importantly, the Roper River is facing considerable water resource development interest, and this is likely to significantly impact our euryhaline species, especially those guys that are going very, very far upstream. We also have commercial fisheries that operate in the Roper River. So we have the mud crab fishery and the barramundi gillnet fishery. So we need to be monitoring these fisheries and seeing the impacts of those on our euryhaline species, as well as doing some further research to understand the potential impacts of that water resource development. So the rest of the talk is going to be talking about acoustic telemetry. So acoustic telemetry involves the use of acoustic receivers, which is the big guy there on the left, and that's deployed and moored in a fixed location in a body of water, for example, the ocean, a river or a lake. And then you tag your animals of interest with one of these acoustic transmitters. I was going to bring one, but I forgot. They're about this big for our biggest V16, which has about a 10-year battery life. So then when an animal is tagged with this, its transmitter is going to be sending out a coded transmission every few seconds. And if the animal swims within range of a receiver, the receiver is going to decode that transmission. It's going to give you a location based on its location, a identification of the animal, and a date and time. So this is the primary method globally for tracking aquatic animals. And as I said, we've got miniaturization of technology, so we can be tracking animals for up to 10 years now. So acoustic telemetry is widely used to understand movement ecology and the drivers of movements. And less commonly, it is used to also accurately calculate natural mortality. So here we have our acoustic receiver array here in the Van Diemen Gulf of the Northern Territory. We have receivers through most of our river systems in the Van Diemen Gulf. In the Adelaide River, which is about 50 kilometres east of us here, we had receivers to 140 kilometres upstream. And then in the South Alligator River in Kakadu National Park, we had receivers up to 100 kilometres upstream. And then we had receivers at the mouths of the Wildman East and West Alligator Rivers to look at any inter-river movements by these sharks. So 200 sharks were tagged in total between 2013 and 2016, and many of those got 10-year tags. So we've got lots of animals giving us 10 years of data. So natural mortality is a demographic process that shapes populations, and it's defined as the proportion of individuals dying from all causes except for anthropogenic mortality, such as fishing. And it can be really used as a measure of species resilience or how quickly a species can recover. It is very difficult to calculate, particularly for aquatic animals, because it's very difficult to directly observe mortality. So as a result, very few species have reliable estimates of natural mortality, despite its importance for the management of commercially exploited species and for management of species of conservation concern. Incorrect estimates of mortality can lead to mismanagement of species. So, for example, overestimating population size or overestimating maximum sustainable yield. So to calculate natural mortality, we can use indirect or direct methods. Indirect methods rely on life history parameters and then plug those into equations. These can be unreliable, and for data poor species, it can be impossible to calculate because we just don't have that data. Often with our indirect methods as well, only one estimate is given. So it's assuming that mortality is constant across size classes and over time, which doesn't make a lot of logical sense. So instead we can use direct methods. So these can be mark recapture, where you're going out, tagging an animal, and then going back on subsequent visits to try and recapture that animal. Or we can use acoustic monitoring. So using our acoustic telemetry, tagging our animals, letting them go, and then our detections on our acoustic receivers are the recaptures. So for our natural mortality estimates, we used data from 185 sharks. These were 46 northern river sharks, 139 speartooth sharks. These animals ranged from neonate, so less than 1 month old, all the way through to adult. And there was large juveniles and sub-adults that were also tagged that would mature within the battery life of the tag. So we did nearly 1 million detections over up to 10 years, and on average 660 days. So the fate of each individual was defined by looking at their detection data, by looking at charts on the View software, which goes along with your acoustic telemetry data, and abacus plots as well. So this is an abacus plot here on the right, and you've just got time on the bottom axis and the receivers going up the y-axis there, ordered in terms of latitude and longitude. And so by looking at all of these plots, we can place animals into categories. So survivals are those animals that made it to within 1 month of their estimated battery life of their transmitter. Potential mortality events where their detections ceased before the end of their battery life, or emigrations where the animals were detected at one of the outermost receivers downstream in the array and then not seen again. So here we have our results of how our animals went for each species and each size class. You can see that our neonates, there was no survivors of any of our neonates. They didn't do very well. And then we have increased numbers of survivals and emigrations once we get into our sub-adults and adults. So then to actually calculate natural mortality, the data was converted into mark recapture histories, and then a function within the fish methods package was used to link and automate the Jolly-Sieber and Kaplan-Meier methods. And this allows for separate estimation of natural mortality and fishing mortality. So as I explained, we've got natural mortality, we've also got fishing mortality, so removal of fish by people, and together combined they equal total mortality. So we aimed to estimate natural mortality, but we couldn't actually monitor for fishing mortality. But given the high usage of lures in the Northern Territory, which results in fewer bycatch of sharks and rays, we also have additional fishing rules in Kakadu National Park, such as the ban on use of bait. And lots of these areas are difficult to access as well. So fishing pressure is considered to be quite low, and we think that these provide reasonable estimates of natural mortality. So here we have our mark recapture histories. Our red crosses are animals that underwent potential mortality events. Our blue crosses are animals that emigrated. And essentially all I want you to take from this is that we have a huge amount of data over a very long period of time. So I really wanted to actually explore the mortality and see if it was differing across temporal and spatial scales. And so to do that, I calculated both yearly and monthly estimates of natural mortality for each species, each size class, and for the Speartooth Shark, each river, because it was tracked in both the Adelaide and the South Alligator River. So what we can see here is just a whole bunch of variability. We have some years where we've got extremely high estimates of natural mortality as well. So looking at our yearly results, our yearly average estimates of natural mortality, this is an accurate depiction of what happens to our baby sharks in the rivers. We have extremely high natural mortality in our neonates. So 0.9 or 90% of our Speartooth shark babies are dying in their first year, and then 73% of our northern river sharks. So this is obviously extremely high considering that we still have mortality in the subsequent size classes. So I did have a look at our catch data from the Adelaide River, and the number of animals caught at age 1 is about 78% lower than the number of animals caught at age 0. So this 0.9 might be a little bit inflated, but it's still around that 80% of our babies dying each year. We then have a significant drop in our mortality estimates as we get into the juveniles and sub-adults, which makes sense. They're getting bigger and they're also getting more experienced. So when we look at our monthly estimates of mortality, we do have a significant difference between size classes for the Speartooth shark. So neonates and juveniles are undergoing significantly higher rates of mortality than their sub-adults and adults. But interestingly, there was no significant difference between size classes for Glyphus garrickii or the northern river shark. But I suspect that if this was to be repeated and more individuals were tracked, that we probably would then get a significant result. And interestingly, again, there was no significant difference between species, season, or location. So despite all of that variation, the mortality is actually quite stable across temporal and spatial scales. So this is the first study to estimate natural mortality for river sharks across size ranges. We have very high natural mortality, particularly for our neonates. And compared to bull sharks, which are occupying a similar ecological niche, we have much higher rates of mortality. So essentially our river sharks are utilising a nursery area that has no refuge from predation. So there's lots of intra and interspecific predation between the sharks. And occasionally our sharks are getting taken by crocodiles as well. But this does demonstrate the value of using acoustic telemetry data for the fine scale estimation of natural mortality, and also is the longest natural mortality study for any shark species. And essentially this study demonstrates the vulnerability of river sharks and that they have very limited capacity to withstand any increased mortality from anthropogenic sources or human-driven sources, such as commercial fisheries or any impacts from water resource development, particularly for those youngest individuals. All right, so our next part of the talk, we'll be talking about movement ecology. And I did this as separate chapters and separate papers for each species. They use essentially the same methods, so I'm just going to go through them together. So for our movement ecology, movement is obviously a fundamental aspect of life, and looking at movement can be used to understand or develop an understanding of ecological processes and implement effective conservation strategies. So for the northern river shark, we had 46 animals tracked in the South Alligator River, resulting in nearly 300,000 detections over 10 years. And then for the Speartooth Shark, we had 143 animals tracked in both the South Alligator and Adelaide rivers, and that was nearly 700,000 detections. All right, so here we're looking at movements over time. So along the x-axis, we have our time, which is 2013 to 2023. And then along our y-axis, we have our distance to mouth. So up the top there, we have 0 and the dotted line. That is the mouth of the river. And then we have 20, 40, 60, and 80 k's upstream. Up the top there, we have stream flow for that river over the same period. This is the South Alligator River. And I will note that in 2017, many of the upstream receivers were removed, which is why the animals suddenly stopped going upstream. But what we can see here is that our northern river sharks are undertaking an annual downstream migration, which coincides with our stream flow. And then they migrate upstream during the dry season. And for our northern river shark, we often have our females extending further upstream than our males. And then if we just look at our size classes, so red is young of the year or neonates, green is the juveniles, and blue is the subadults and adults. We can also see that our young of the year are extending further upstream as well. So we have both size and sex segregation in our northern river sharks. And we also see that our subadults and adults start to spend more and more time closer to the mouth and not extending so far upstream. For our Speartooth Shark, we have size, age at size estimates. So we could place each animal into an age when they were released, and then they had a birthday every year on October 15th, which is when they're pupping. So these different colours are those age groups. And then at the top, we have our water level and our stream flow for, this is the Adelaide River. So again, we can see this really clear annual downstream migration during the wet season, coinciding with our periods of high stream flow. And then they're extending up into upstream in the dry season. And so for our Speartooth sharks in the Adelaide River, sharks are generally between 60 to 100 kilometres upstream in the dry season. And there was actually 4 sharks that were detected all the way to 140 kilometres upstream, but they were in the late dry seasons of 2019 and 2020, which, if you remember, were just after years of bad wets. So when there is less freshwater flow, they're actually extending further upstream. And again, with our Speartooth sharks, you can see that our adults and our larger groups are really starting to spend less time upstream as they age. So unlike our northern river shark, we really don't see any indication of size and sex segregation in our Speartooth shark, which is quite interesting. All right, so space use. Overall and monthly activity space of individuals was calculated using Brownian grid utilisation distributions, or BBUDs, to identify areas of higher usage by each animal. So the extent of area use, or 95% BBUD, was examined over monthly intervals to explore changes in movement and space use over time. So movement paths for the calculation of BBUDs were calculated using least cost paths. So the water was made to be cheap and the land was made to be expensive, so that our paths would follow the rivers and not through land. So that's how we get this nice thing, but there's a few places where still just really want to skip over land. And then our BBUDs were calculated using SPATStat to using a spatial area of 200 metres based on our receiver range and increasing with 5% with every hour of transit between detections. So here we have our space use for the northern river shark. Our hexagons are just locations we have each month of the year for the river, and then the number of individuals occupying that space in that month is the colour. So where we have red, we have lots of individuals, and where we have blue, we have less individuals. So we can see again in the dry season, sharks are upstream. They're usually around 50 to 80 kilometres upstream in the South Alligator River. 12 northern river sharks did extend to at least 100 kilometres upstream. And then in the wet, we have our sharks going downstream and using the mouth and actually moving across to the West Alligator River as well. So they're usually occupying the space around the mouth to about 10 kilometres upstream. Here we have our space uses for the Speartooth Shark for both the South Alligator and the Adelaide River. Again, we're seeing that clear upstream usage during the dry season, downstream usage during the wet. So we have peak downstream space use occurring in January to March, and then peak upstream use occurring towards the late dry, so around October, November. So I did use best fit candidate models to look at the significance of environmental factors on the space use. But when I did this, I put stream flow, rainfall, and air temperature, which was the only thing that we had for these areas. But those are all obviously linked. So I'm going to have to do this again and probably just use stream flow. But essentially, our stream flow is having a significant impact on our space use, where the space use is actually decreasing when we have increased stream flow. So that's probably because they're all getting together around the mouth, occupying a smaller space, but there's also probably less receiver coverage of the mouth as well. So if we think about why these animals are doing this, this is a lovely graph from one of the data loggers that Erica Kalini and I put in the Adelaide River. And so when we have this influx of freshwater flow, which triggers the sharks to move downstream, we have a really significant drop in our specific conductivity, which is really closely linked to salinity. So these sharks can occur in freshwater, and they do occur in freshwater, but they prefer a certain amount of salinity. And so in the wet season, these guys are actually following that salinity out to the mouth, where they're going to have a little bit more marine input. We did have inter-river movements by some animals, but it was very few. So most of our sharks are remaining in their natal rivers, and there's very few that are actually moving between river systems. So for the northern river shark, only 5 individuals out of 46 moved between rivers. These were 2 females and 3 males. And these movements all occurred between the South Alligator and the West Alligator in the wet season. So they're moving about 19 kilometres. There was one shark that did it in less than 24 hours, going back and forth, like going to the west and then coming back. One female did move between the South and the West Alligator River 12 times between 2014 and 2019, all in the wet season. And she did go over to the Wildman once in 2015, which is about a 50 kilometre swim. Speartooth sharks, same. There's only 10 individuals which did inter-river movements. 7 of these were females, 3 of these were males. So we did have animals tracked in the Adelaide and South Alligator River. And again, they stayed in their natal river and swam between rivers before either emigrating or returning back home. So our animals from the Adelaide River swam over to the South Alligator River, skipped all the rivers in between, 175 kilometres. They did this in the dry season, and these were older individuals, so about 10 to 17 years old. And then our sharks that were tagged in the South Alligator River, they again swam between the south and the west before returning back to the south. And these were happening in the wet season, and these movements were occurring in younger animals. So pretty interesting inter-river movements there. So this is my favourite shark. I did not have time to animate it, but here we go. This was an individual tagged in 2016. She was a female speartooth shark. She was approximately 10 years old at the time of tagging, and she was 176 centimetres total length, so big girl. She spent about a year moving normally throughout the Adelaide River, up and down, and then she left in April 2017. She then swims all the way over to the South Alligator River, but she takes 2 years to do that movement, so she's moving somewhere in the coastals. And she appears at the mouth of the South in March 2019. She then disappears again until May 2023. And then she pops up in the Wildman River, so that's after 4 years. She then leaves the Wildman and swims over to the Adelaide River in late August of 2023. And so in late August, we're starting to get into pupping season. So this potentially is a girl who has returned for the first time to give birth back in her natal river. And she was about 16 years old when she came back to the Adelaide. And then she swims back to the Wildman River in January, and then she disappears again forever. Because we took the receipts out, which is so sad. All right, so we have very high river fidelity. These guys are really choosing to stay within their natal rivers. And we also have extensive use of river systems by adult river sharks, which we did not expect. So speartooth sharks are understood to mature at about 12 years old, at which point we thought that they would immediately leave the rivers and start living their life in the coastals. 27 sharks were tracked aged 12 and above for our speartooth sharks, but only 7 of these undertook inter-river migrations or emigrated. So that's only about a quarter of them. The remaining 20 stayed in their natal river systems, including 3 16 and 17-year-olds that had not yet emigrated at their last detection. So that was a very surprising find. We know that annual migrations are strongly driven by the environment, primarily that freshwater flow. And these movements have management implications. So what is water resource development going to do for these migrations? We have spatial and temporal overlap with fisheries. So, for example, we've got all of our sharks moving downstream in the wet season, and then we have the barramundi gillnet fishery starting up on the 1st of February every year. So at that time, we've got heaps of sharks hanging around now. So there is the potential there that we could advocate for a seasonal closure and just push back the opening of that fishery. So what have we learnt? Euryhaline species are unique, threatened, and very poorly understood. The Roper River is an important area for threatened euryhaline elasmobranchs, including an important reproductive area for speartooth sharks. River sharks undergo extremely high rates of natural mortality, severely reducing their capacity to sustain increased sources of mortality from humans. We have seasonal migrations in response to the environment, and all of these have implications for their management. So what do we need to do next? In Queensland, in the Wenlock River, a study found that 1 neonate shark per crab pot per day was being lost as bycatch because they are small enough to swim into crab pots when they're first born. So we urgently do need to find out whether this is occurring in the Northern Territory, because that's another situation where, again, we could close the fishery or close the crab pot fishery around the time that we're having our pupping season and stop those neonates from being lost so that we don't have increased neonate mortality. We can, yes, improve our monitoring and management of fisheries to ensure that we're not losing too many of these species to bycatch. We can use all of this movement and mortality data to improve the management. So again, looking at that water resource development in our fisheries. Oops. We are still lacking a lot of life history data for these species, but particularly for the northern river shark, which pretty much is completely unknown in terms of life history still. Data from my 105 speartooth sharks from the Roper River is being analysed using close kin mark recapture, so essentially looking at how related each sample is, and you can use that to figure out the abundance, so how many reproductive adults we have in that population, as well as looking at their connectivity. And we're especially interested in connectivity between the Roper River and the Wentlock River on Cape York in Queensland. And we still have completely no idea where our adults go when they leave the rivers. And so we don't know how they're using marine habitats, what habitats they are using, and we don't know how much spatial overlap they're having with commercial fisheries. So satellite tracking is a key next step for our adult river sharks. If anyone knows how to find such funding, I'd love to know. But yes, thank you to all of my advisors, supervisors, co-authors, all of the field volunteers that came before me and during my time as well. Thank you. Any questions?
Julia, any questions? First, we'll take some from the room.
I was wondering, you found that like consistent movement down and upstream throughout the year. Do you think they're just being pushed downstream by the higher flow, or do you think they're using different resources?
So they are very strong swimmers and they're very fast. So if they wanted to swim like against the flow, that they could, but they would certainly use a lot more energy doing that. But you certainly don't see this kind of movement really that clearly in other species living in the rivers. They might often actually push up into wetlands and things and use all of those wetland resources when it floods. So this is quite a unique river shark thing that they're doing. Yeah.
There's no idea sort of why they would.
It's going to be from that salinity, all of that freshwater flow pushing, bringing down the salinity, and then they're going to go to marine and get that marine-driven salinity out there.
That's wonderful to see so much data on such rare species and effectively what you would call pre-impact data.
Yeah.
Because I was wondering if you know if there are any plans to keep the Adelaide River study at least going with the Adelaide River offtake scheme, because it seemed that that is a perfect indicator if you've already got that pre-impact data.
Yeah, so it was something that was discussed a couple of years ago, but essentially you would have to start again from scratch. You would need to go out and tag 200 more animals, and you'd have to redeploy all of the receivers. All of our receivers were getting pretty old, so I did recommend that we would replace them, or at least replace most of them. I think that that's kind of fallen off the radar because people realise that it's actually quite a challenging thing to do. But certainly it is something that we should keep in mind, that it is certainly a good before and after experiment that could be done. It should sound like. Yeah.
With just around the idea of water resource development again, like what do you think are the main risks that we need to know more about? You know, is it changing salinity impacting where they can actually hang out, or amount of fresh water and food availability, or all the above and other things? Like
it's very tricky. We don't know how it's going to impact them because potentially it will stop that downstream migration. But is that a good thing or is that a bad thing? Will that affect other parts of their behaviour and life history? Potentially the removal of that fresh water is actually going to create more habitat. So we saw in that 2019-2020 after those bad years that they were extending further upstream. But the main problem is going to be that we have all of these trophic cascades, you know, food web. So these species really heavily rely on riverine sources of prey, and they actually have, looking at their fatty acid analysis, they actually have diets that are more similar to rays than to sharks of their size. So they're actually occupying kind of lower trophic levels. So we know that freshwater extraction is going to be very, very bad for our fish populations, and so we're going to have essentially just a catastrophe of these guys not having enough food. But it is certainly something that we don't really understand how they react.
We will take one from the online.
Yes, this is a question from Ross online. He says, what is it about the Adelaide, South Alligator and Wildman rivers that are attractive to geographers compared to the rivers that they seem to avoid? Is it true that avoidance of these rivers follows the fact that there were no receivers in the other rivers?
So the only river that is between, so it essentially goes the Adelaide, the Mary, then the Wildman, the West and the South. So there was no receivers in the Mary, so we don't know whether they did pop in there. But then there is always the potential that sharks just weren't picked up. So in these river systems, we actually have terrible acoustic receiver range. So on a good day, when it's nice and clean and we're on the neap tides and nothing's really moving around, we got about 400 metres. So as in the shark has to swim within 400 metres of the receiver. But upstream in the Adelaide River, where we're talking really, really muddy, noisy water, spring tides, we got 20 metres. So that literally means the shark's got to swim right next to the receiver. So there is the potential that sharks weren't picked up in the Wildman and the West. But it certainly was, for whatever reason, they're just heading straight to that next biggest river and then going back home. But in terms of why they would do that, we would have to do more research. Maya, yeah.
Oh, it's like interesting because in Indonesia, river shark mostly only a story. Then we just like found it in fossil or whatever, but it still exists here, and it's like really important research, I think, for conservation. And first, it's like about the natural mortality, because mostly it's interesting because mostly the juvenile shark, it has like higher probability to survive, but river shark is not. Like, do you think it's like because of the prey, or like what is their food mostly, or they are eaten by crocs mostly, or what is the story behind that? And that's
It's definitely not mostly crocs.
yeah.
Across other species that I looked at for natural mortality. So generally it's the juveniles that undergo higher rates because they are smaller bodied. So obviously you get bigger, there's less things that can eat you. There were some species that was around the 80%, but then some species that were lower, like 50% or lower. I think with these guys, they're a pretty small body species. So for example, last year I caught a bull shark. A freshly pupped bull shark still had a bit of an umbilical cord attached. So it literally just been born in the last couple of days. It was the size of a 1-year-old Speartooth shark. So these guys are just really small. And so when they give birth, they're pretty fat and they're pretty happy because they've been supported by their mum. But they've really got to learn in those first few weeks how to hunt. And you actually find that a lot of the neonates get really skinny. So some of them just maybe don't quite figure out how to do it and what to do. But certainly when we're going fishing, a lot of the sharks have pretty serious bite marks from other sharks.
And then is there any like relationship between elevation of the river? Because last year when we went to Indonesia, we found Gripis Gangeticus in Borneo, in Kalimantan, and then it was in the highlands. Is there like any probability they swim upstream than, like, you know, with the high elevation?
I don't know anything about elevation, but for these guys, it's mostly going to be driven by substrate, whether they're there, because they really do like that turbidity. So they're probably not going to go so much into rocky, sandy rivers where it's cleaner, but they will go into those muddy, fine substrates. But yeah, I couldn't tell you about elevation. Yeah.
Across your like longitudinal data set, did mortality of neonates or adults change through time, like with the seasons?
Yeah, let me go back to our giant table. Essentially our neonates all died like very quickly within the first year. But if we look at these data, it's kind of variable most years, but our sub-adults for both species, they do become almost zero for most years. I think we can ignore Glyphis garrica because they have a really high mortality here in 2016, and that brings up their, in 2014, I should say, that brings up their average. But if we look at, our Speartooth shark that we have lots of data for, it does drop quite significantly as they become older and older.
Yeah, sorry. I'm on that, like, with poorer seasons in 2017, 2018.
Yeah.
Was mortality?
Well, there was no difference between seasons, as in, because I did think that there would be more mortality in one of the seasons at least. I think I thought there would be more mortality in the wet season, because they're all kind of grouping together, and then there's shark on shark stuff going on. But yeah, there was no significant difference there. So it's something that we'd have to look at further. And there's a lot of stuff that you can then pull from this and go explore. You had the funds.
Good. Next to Jenny.
Just more about methodology. Did you, when you were doing this testing and trapping, particularly as we're talking about the neonate mortality
Yeah.
conversation, the difference between the audio telemetry and the marker capture.
Yeah.
I know they kind of distinctive.
So we didn't do marker capture, we just did the acoustic telemetry. So we had V16, V13s and V9 tags. So all of the smallest individuals got a V9 tag because it's a lot smaller, it's going to have a lot less impact on them physically. And then also any deaths that happened within the first 24 hours were excluded because they could have been the impacts of tagging. So we certainly tried to reduce the impacts of tagging on these.
But you don't expect any even kind of like longer than 24-hour impact.
There is the potential, but 24 hours to 48 hours is generally considered enough time for them to recover. There was a couple of really weird individuals that I kept out as well. For example, they just disappeared for 6 months and then reappeared somewhere completely different. So there was a few where I was like, is this being eaten by a crocodile instead? But yeah, essentially what I could do to bring this conservative, I did, yeah.
I guess I meant rather than risk of the handling, I meant more perhaps the longer term impacts to this communication.
No, so this acoustic tagging, it's done internally. They have a little surgery, they're anaesthetised, they get stitched up, and actually one of the sharks was recaptured a month later and you couldn't even see, like you could just see a fade line. So sharks and rays do have extremely good immune systems, they recover really well. So there shouldn't really be anything long term apart from natural effects.
I have time for maybe just two more if they're quick. Are there any more online? Okay.
Just with the death of all the neonates, you mentioned it's mostly, you think, natural mortality. And so it's really cool to see all this data for like really threatened species or species that are data deficient. But if you're trying to like think about that in a conservation from a conservation standpoint, if there's no real human impacts on that really low mortality, how do you then try to increase the survival of the neonates to try to lead to like larger populations?
Yeah. So we know that this is we know that they're not doing. I mean, we know that our babies don't survive very well. But in terms of river sharks elsewhere, so Naya talked about Glyphus nianticus, they are doing very badly because of overfishing and effects on their habitats. But in Australia, we really don't have we're not so worried about our river sharks here. So there's not anything that we would do to try and increase their natural survival, but we would just try and make sure that we're not increasing their mortality because of us. Yeah.
Do you have any sort of recommendations to the recreational fishers as to report what they get? Do you have anything?
I think that there was a time where people were being asked to report what they were seeing. Unfortunately, in the Northern Territory, we still have recreational fishers that can't tell the difference between bull sharks and river sharks. So a lot of people, when I talk about river sharks, they'll be like, oh yeah, I caught river sharks way up, in the escarpment country, wherever they've gone fishing. And I'll usually be like, no, that's probably a bull shark.
So it's misinformation.
Yeah, so, and a lot of people aren't as you saw with the Roper River, we're actually in quite a narrow stretch of the river that they're actually occurring. In the South Alligator River and the Adelaide River, they're obviously going a lot further and they're going to have potentially more interactions with recreational fishers. But yeah, I think the first thing to be with recreational fishers would be to educate them on how to identify them and why they're important and why they should chuck them up.
Because I was surprised on the Adelaide River how many people are fishing recreationally.
Yeah.
This is sort of near the bridge and where you go.
Yeah, so there's but for the Adelaide, there's like really only those few main sites like Maracay Creek around the bridge. When you go further towards the north, there's pretty much no one that goes up there or very few people. But yes, we can certainly do more education around that goes. Yeah.
Last one.
When tagging the shark, why do you tag it exactly, like precisely? Like, does it differ according to the species, like each and every shark? You tag exactly in the same way, like any measurements or something like that?
Yeah, so pretty much when we catch a shark, we will take measurements, we'll assess their maturity, we'll give them a pit tag, which is essentially this tiny little tag that goes in, we put it on the right side of their caudal fin in a muscle. So that's always standardised so that when we recapture a shark, we know where to scan to try and find the tag. And then there is just best practice ways of doing things. So for the acoustic tags, you can tag them externally so that you're not having to do the surgery, but then you have higher chance of losing the tag and also biofouling and things, and you'll affect the drag of the shark as well. So you'll increase the amount of energy that they're having to use to swim. So the internal tagging is, a very standardized process that pretty much everyone uses.
Right. Well, join me in thanking Julia one more time. And please join us in 2 weeks' time when we have our next RIEL seminar. It'll be Andrew Harford. He's from the Office of the Supervising Scientist, who will be presenting. And also, please, you're welcome to join us. We have our RIEL morning tea that starts at 10:30 in the second floor of Yellow Tube, the next building.